Hello, everyone. Welcome again to the BugSpeaks podcast. In today's podcast, we're going to talk about something very new and something very different called as fecal microbiota transplant or FMT. FMT is a fairly new technology that we're seeing, but it also has a lot of historical importance as well. And we'll talk more about that today. As usual, with me, I have Dr. Debojyoti to discuss this. Deb, your first thoughts on FMT?
We are touching on a topic, Kumar, which is very cross, but I think in the microbiome field, this has to be discussed. As you rightly mentioned, FMT is fecal microbiota transplantation. Although we are talking about it now, but it has a very old history. If my memory serves me right, uh, way back in 4th or 5th century, it was the Chinese who basically took the fecal matter, made a suspension out of it. I think they used to call it a yellow soup or a golden soup, and they used to treat some of the patients who had diarrhea and orally. So, so that was quite gross. But now, with the passing few, in fact, in the 1950s, there were some trials done on FMT in properly in our medical domain clinical trials. Um, but now FMT has become a very important tool in the hands of physician to as an experimental tool, if I can use that word, uh, to address certain unmet medical needs. So in this podcast, we are going to discuss, I think, most of those uh, domains where FMT is being used. We are also going to discuss about the issues with FMT and the trends that we see in the FMT. So moving forward, Kumar, I mean, as you started this podcast with this three word sentence, FMT, fecal microbiota transplantation. Um, FDA has approved FMT only for a certain disease, which is C. difficile infection. Your thoughts on this particular aspect?
It is surprising that it's been approved only for one disease, whereas you look at the Australian FDA counterpart, they've kind of approved it for much more diseases. The last time that I checked the CTRI portal, not the CTRI portal, the global clinical trial portal, there were, I think, about 130 diseases for which FMT is being currently tried for. So everything from gut-related issues to boildness, I think FMT is being touted as a possible solution. But uh, I'd like to focus on the fact that there is a rigorous trial that is required before an approval is got. I mean, this is in fact a new technology. We still don't know what are the full ramifications of doing an FMT. Till now, we don't see too many side effects. So it's very rare. But at the same time, it's better to get a clinical trial done for many diseases to get to know the standpoint. When I was talking to a couple of doctors about FMT, and I was surprised to know that even in India, in between the 1930s and 1940s, I mean, there were a lot of, especially South India, I've heard that there was a lot of FMT usage.
Yes, that's interesting.
CMC Vellore and some of these places, there were people routinely practicing FMT at that point of time. Maybe not the same way as it is done now, but maybe it was the yellow soap kind of method in which it was done. Um, in talking about the FDA approvals, I mean, I'm sure you've come across a few studies where FDA has been going through,
Sure.
and if you can throw some light on that as well.
Sure. But before that, Kumar, I think let's elaborate on FMT for the audience. We have not done that. So FMT basically is fecal microbiota transplantation. So as the name suggests, what happens here is uh, the fecal matter is taken from a donor, a healthy donor, and transplanted into a recipient. So this basically is what FMT is all about. Uh, the transplantation happens mostly through the rectal route. So it sounds very gross, But nowadays, people are working on other modes of transplantation. A nasogastric route is another one where they put a tube through your nose to the through the esophagus, and it goes to that alimentary canal and all those things. Then the frontier is also on the oral capsules, FMT capsules as we speak. Now coming back to your question, Umar, about the other areas where FMT is being tried. I think any disease that you can think of, chronic disease, starting from cancer to NFLD, which is nowadays called as MFLD, uh, to autism, autism spectrum disorder, depression, Parkinson's. Scientists, clinicians are trying FMT in all these diseases with varied results. When I say varied, apart from the C. difficile and infection, the FMT success rate in C. difficile infection is more than 85%, especially for recurrent C. difficile infection. Whereas an average success rate of around 50 to 60% is for the other diseases combined. I mean, so there will be certain diseases where the success rate might be even higher, and certain diseases where the success rate is lower. But although it sounds gross, but diseases like these where generally people hit a wall, it comes as a savior. So there have been many instances where FMT worked well. Generally, FMT is done in certain sessions. Some people recover after first session. Some people take 3, 4, 5 sessions, maybe depending on the disease and how their gut initially is. We'll be talking about, Kumar, the various parameters that are looked into getting a proper FMT done. But because there is variation in so many factors, starting from donor selection to the genetics of the recipient, there's variation in the success rates also. So, so let's talk more on those variations, since we touched upon that. How important is donor selection? If you can shed some light on that, it will be great.
That was a most interesting thing about the FMT journey as we were looking into it. Most labs report that they screen at least about 100 people before they get a successful donor. And when I say screen, I mean they're screening for blood parameters, for looking at any pathogens that are present. And this is after screening thousands of people on questionnaire based methods. So putting all of that together, getting the right donor is very important.
Right.
I remember when we first wanted to do a FMT in 2018, the first doctor who we met suggested that we should go to gyms and collect the donor samples. And as we've done so many athletes and people working on strenuously in gyms, we've checked the microbiome. We realized that their bodies are under so much of pressure that their microbiome is actually disrupted, and they're probably the worst candidates for FMT. So donor selection is not easy. There have been theories that you take a person from a village, put him in an isolated setup, give him good food, and you'll get the perfect donor. But none of this is practical and does not work. There are amount of tobacco usage, the um, amount of alcohol that they might be using, the kind of food that they might be eating. In India, what is considered as medication is very varied, right? I mean, most people don't think crocin is a medication. No, oh, um, बुखार की दवा है type. So it is not like a medication. So in the questionnaire, when you're saying, are you under any medication, the answer comes out as no. So getting all this right and getting to the right donor is difficult. Few of the technical parameters that you can talk about in the donor selection.
Yeah. So you've highlighted, I think, many, basically the blood parameters, mostly the infectious, agents that might be lurking in the blood, in the stool, um, like, the general pathogen Shigella, and which might cause dysentery and all those things. So those are generally done. Uh, uh, then the history of that person. I mean, the person, has he ever taken antibiotics? If he has taken antibiotics in the last 30 days or 50 days, that person is not eligible for the being the donor. Uh, no smoking, no alcohol. But you know what? From our understanding of the microbiome space, we've realized that just based on those parameters, uh, selecting a donor is not the right idea. Now that we have seen so many uh, gut microbiome samples, we have we have now understood that having a good gut microbiome may not be completely dependent on you not smoking, or it's a combination of so many factors, right? The donor has to be really, really, when we call fit, it's not the external fitness that we are talking about, it's the internal fitness. And internal fitness, there are so many variables, starting from the lifestyle, the habits that you have, and also your genetics. So there are a lot of research, Kumar, you would know, that people have found that uh, the term that we are going to use in this episode, like colonization, which basically means how the organisms go and reside in the recipient's gut. There's a term called colonization potential. So research has suggested, again, this is an ongoing field of research, nothing can be absolutely said at this point of time. But presence of certain organisms in the donor sometimes dictate how well the ecosystem is going to go and reside in the recipient's gut. Similarly, the recipient's gut microflora also plays a very important role. You might have the best of the donors, but if your recipient is not, the gut is not in a very good shape in terms of the ecosystem that that person is having, or the genetics part of it, which is still under active research, then also there is a potential that the colonization will not happen. So many variables are there. So our thought on this is mostly that the questionnaire will give you a first cutoff, but ultimately it is the microbiome that we need to really look at, and that I think that is the trend that we are going to observe in this domain as we move forward. So talking about trends, Kumar, what are the trends that you see in the FMT space apart from the donor selection that might include the metagenomics screening? What else do you see in this trend?
See, there are three hotspots of FMT worldwide. One is Boston, the other one is Hong Kong, and the third one is Australia. And I think Australia is much advanced than the other two. Professor Borodi of Australia, who unfortunately recently passed away, was a pioneer of this in this field. I mean, he's done so much of meaningful research. And from all these people, we're learning facts about donor selection. We're learning facts about how the microbiome is also playing an important role. Don't only look for infection. Your favorite subject, how it's not only bacteria but it's also the fungus and the phagus that's in the gut that's playing a role, and that translates to better FMT success. And FMT, what is now coming out, what was a often child before and never, not often neglected child before, was all the metabolites that's actually there in the stool sample. It's the products that the microorganisms produce more than the microorganisms themselves that could be acting as a good source when the transplant happens. The second type of trend that we see is the number of sittings has gradually come down. Usually, it used to be 6 to 8 cycles of colonoscopy through which FMT was done, but now you're repeatedly hearing that the number of cycles have come down to 1 to 3 kind of cycles, and I think that is good. Though the worrying thing a little bit is that there are some reports that are coming out which says that once the FMT is done and the patient, if the patient does not take care of him or herself, then in a year's time, the disease kind of recurs and they'll have to go back to FMT. So FMT is one thing, but after that, it is important to look after your health and make sure that you don't fall ill again. Kind of thing. So, what are the necessary precautions that are there? So, FMT is evolving. I think we're in a cusp of becoming FMT 2.0. But at the same time, I think we need to acknowledge all the issues with the FMT as well, and which is why the clinical trial is important. If you can address some of the issues that you would see.
So the greatest issue or the biggest issue is the variability. And the variability starts, see, biology, as I always say, is a science of exceptions. Unlike physics, where it's a science of rules. And because you are dealing with humans or any organism, there will be variation starting from there itself, right? So issues with FMT, Kumar, one of the major issues is donor selection. Because nowadays people are trying to not I will not say nowadays, but people have been trying to look at donors only with respect to the questionnaires. They were not looking deep into the microbiome of that donor. Second issue with FMT is we still do not know, because it's an ecosystem of organisms, uh, and it's undefined, right? So we still do not know uh, which organism is doing what. The cohort of the organisms which is going from one person to another, how it is impacting the host is something that we still do not know. You touched upon the viriome part or the viruses part. Interestingly, people have also shown that when they filter out all the bacteria and fungi and other filtrate from the milieu, what remains is the phages in the milieu. Even that milieu with the phages, when transplanted, did what the complete ecosystem was doing. Not in all cases, but in some cases they have found. That means phages are playing a very important role. We mostly focus on the bacteria because that's the most important or the abundant kingdom in that ecosystem. But the fungi and the phages also make a lot of sense. So, and the third is, of course, the issues in terms of regulatory issues, because this is something which the regulatory agencies are also keeping a tab on, because it's an experimental tool right now, and a lot of trials are happening worldwide. I think they are also looking at uh, trying to evolve with times and see. One small thing I would just add here is during COVID, it was done. I mean, because FMT was being done during that time also, maybe. And there was one case where someone got infected because we know that SARS-CoV-2 virus is present in the stool of COVID-19 patients. So they have now mandated to screen COVID-19 or SARS-CoV-2 in the stool also, which was not there pre-COVID. So people are evolving, the regulatory agencies are evolving, but we haven't reached that stage where FMT can be used as a definitive therapy for a particular disease, except C. difficile infection, which has been approved in US. Uh, I would like to touch upon another aspect, Kumar, which is the miniaturization of FMT, if I can use that word, like using the oral capsules. Uh, why don't you talk about the two FMT products, Rebiota and Voust, which were recently approved by FDA, because that's something for the microbiome space. It was a shot in the arm. So much of investments have gone into this domain. So if you can take the audience through to those two products and how the next FMT 2.0 will be, it will be great.
Sure. So let me zoom out a little bit over there for touching upon this topic. I mean, so there are two different aspects as to why this evolved. First one was FMT as a procedure did not have any IP behind it. So there was no pharma company willing to take a lead to spend millions of dollars to make FMT as a prominent technology, go through all the clinical trials. So because of that, there had to be innovation that had to come about where IP can be brought into it. Otherwise, the R&D spend was going to be a little difficult. Secondly, the process of FMT itself is cumbersome. You take the donor stool sample. If you process it too much, you can increase the shelf life but decrease the efficacy. If you don't process this, the shelf life comes down but the efficacy goes up. So there was a little bit of a mismatch over there, and problems of, say, hitting it up and getting getting the right sample to the right recipient was becoming a problem. And lastly, donor selection was so cumbersome that once you got a donor sample, it was like gold, and you wanted to make the maximum out of that. And this is where I think all these forces combined led many biotech companies and then pharma companies to look at how do we productize FMT. One, to make the efficacy much more sustainable, and secondly, to improve the cost of R&D and the outcomes and all that. Only then will we get a world-class product. And this was done through making sure that the essence of the FMT remains, but you'd be able to put it into a capsule. So basically, you're taking the stool sample, um, in a way making it into a powder so that you can put it into a capsule and then giving it to the patient. So that was the thought and the force behind coming together. But you know more about the clinical trials that were done around this, and I think if you can touch a little bit on that.
So yeah, I mean, see, these two products that we just mentioned, the Rebiota is from Ferring Pharmaceuticals and Voust is from Ceres Therapeutics, and both have been approved for C. difficile infection. The difference between these two, and we will see the evolution here itself. Rebiota is an enema-based, product. It's a solution basically, and it's an enema-based. So that means the patient has to be there in a hospital setting, and a healthcare giver has to give that enema or something. Whereas Voust is an oral capsule. Also, there is a subtle difference in the constituents of it. Voust is more of a defined kind of a consortium. They're heading there, whereas Rebiota is a lesser defined consortium. I think the trend there also is to move towards more defined consortium of organisms in the capsule that can be put, so that see, ultimately, as scientists, as clinicians, no one wants to put an undefined thing in a patient, although it might work. But the emphasis now is to understand how it is working, why it is working, and which are the organisms which are making it work. Again, it's a very complicated, complex interplay of organisms. It's probably it is very difficult to pinpoint on one or two organisms, because there are trillions of organisms and thousands of species. But having said that, again, it is an endeavor as scientists to understand which are these microorganisms or species which are working together to give an effect that we see in FMT. So that's how these two, are working. I think there are a few more companies that are working. For example, there's a company called Mat Pharma. It's an European company. They're focusing mostly on the immunological aspect of it. I think they recently did a phase 2 or phase 3 clinical trial on graft versus host disease, and they have got good results. Fingers crossed. Once they really see and show good outcomes, maybe the regulatory authorities will look at FMT derived products as the next frontiers in treating disease. The big names in the pharma are also very interested, as you know, Johnson and Johnson, GSK, Nestle Health Science is very much into the game. I think they have tied up with Ceres Therapeutics to market Voust. And of course, there are other companies in this domain like Finch Therapeutics, Vedanta. They have their late phase clinical trials happening for different diseases, including C. difficile and of course IBD and cancer and so on and so forth. So yeah, so this field is evolving, and there's a lot of interest from the big pharma and the investor community. What we need is good products, and then I think this FMT derived product category will also go alongside the FMT, the normal stool suspension FMT. That also is evolving in its own space. So Kumar, to conclude this session, what is your take on FMT in India, and where does BugSpeaks stand?
FMT in India, I think AIMS is starting to do a trial on FMT in India. I think donor selection is happening for that. At least a few months ago, that was the status. That was one of the trials. Apart from that, in general, FMT is happening under the radar. And it's a bit of a hit and a miss because it's not been done too scientifically. and no proper clinical trial has been done. So in a very hush-hush manner, there are a few hospitals that have been doing FMT. And I mean, talking to clients all day, I kind of understand why this has been happening, because a lot of clients want FMT done and they just cannot afford to go to any of the international centers to get it done. So there is a demand for FMT. What we wanted to do was to, with BugSpeaks, wanted to make it as scientifically thorough as possible. I think we had a couple of missteps. Started once in 2018 to try and do FMT. It didn't work out. Then try one more time. So I think this is like the third time, but we've learned from those mistakes. The first thing is we've got a good clinical partner to partner with us. I mean, so there is a hospital that can execute all the things that we want to do. The first thing that we're going to do is start a clinical trial. I think all the registration for that is happening right now, and we'll be starting a donor selection pretty soon. So if you think you're a good donor, please donate some some samples to us. So donor selection will happen soon, but we want to go through that clinical trial process, make sure that it is there is merit to whatever method that we're following, and the solution that we're going to give. So that's the step that we want to take. And I know you wanted to add a zing to the product. It has to be a good differentiator.
Of course. Yeah, you have rightly, put that forth. For us, donor selection with a BugSpeaks touch has to happen. It's not that just that we want to, we want to look at the microbiome in a very different manner. I mean, we are developing something called as BugSpeaks FMT score as we speak. The endeavor is that can we predict based on the score that we develop that a particular donor is going to be efficacious for the FMT. Again, as I said, there are a lot of variables. We cannot control all the variables, but at least if you can have some control over one of the variables, which is the most important variable maybe, which is the microbiome, I think then the efficacy of FMT can be improved upon. And as you rightly said, we are going to do that in a clinical trial route, because without clinical trial results, we should not claim anything for the public. So we believe in that. Otherwise, there are so many clinical trials we as a small startup we have been doing is only to show that the science works.
Yes.
And that's how we are going to do it. So with this, I think we have come to the end of this episode. I hope the audience understood a little bit about FMT. The gross nature of the technology or the therapy is not important, but the efficacy of it is very important. Thank you, Kumar, for your time, and we'll see the audience in our next episode. Thank you. Bye.
Thank you.